Welcome back. So, I just spent probably 20 minutes. This is a longer lecture. 20 minutes recording. I was in my bag, explaining things super awesome. Looked down, the record button wasn't going. Talking to myself for 20 minutes of this lecture out loud. Crazy person. So I'm a little perturbed. But we're going to do it again. I see it recording now. Okay, so I don't know. I'm just out of my mind. It's like a video confession. None of you are old enough to probably remember The Real World, which is like an MTV show. But they had like this video confession booth where they just went in and like just bitched about other people or whatever. That's what I feel like right now. I'm just letting it out. All right, let's do it. Somitegenesis and somite development. So here's our Bob Ross style picture of a somite. These happy little trees. Another thing is probably, I just date myself. I just feel old when I make these lectures or talk to you guys in class. Anyways, you probably know who Bob Ross is, but like in an ironic way. All right, let's go. All right, somites. So here we have somites. These are these little like bundles of cells here, or kind of like units. This is a neural tube down the center here, and this is an electron micrograph where the surface ectoderm is peeled away, and we have these well-formed somites that are revealed. So those are the well-formed ones. Down below it, we have the paraxial mesoderm. This paraxial mesoderm is not yet segmented or fully developed into these somites. So this is kind of the process of what's going on here. Okay, man, I explained this all so good last time. Now I'm gonna probably bungle it up. There's a lot going on here. So what I'm going to start with is we're going to define some of these things because it's like alphabet soup of acronyms here. So first, NC, Notochord. You guys know that, right? We also have the DMZ, which is not the demilitarization zone, like in between the Koreas. It is the dorsal medial zone, all right? We also have NMPs, which are neuromesodermal progenal cells. Progenitor cells, not progenol cells. Progenitors right here. Okay. Now, I think that's maturation. Okay. All right. So what's happening here? So we have there's three processes that lead to the elongation of the notochord, and as it's elongating, development of these somites along the way, right? So, or elongation of the neural tube, I should say, which is pulled by that notochord. All right, so up here, we have these NMPs, remember, neuromesodermal progenitor cells, and they collect up here, and then they migrate. And they're bipotential, means two potentials, right? So you can either move into the neural tube and become neural, or they can move to the PSM through this maturation zone and the progenitor zone and become mesoderm cells, right? These progenitor cells, as they move, express the CDC25A+, quite the gene name there. What that does is it causes them to start to divide, right, and proliferate. So you can see that's kind of very finely shown with these spindle fibers and these dividing cells here. All right. Now, that causes this growth. Dividing cells causes growth, right? In addition to that, we have, so this is laying down more of this mesoderm, which will become somites, right? In addition to that, we have the notochord, and within the notochord cells, we have these vacuoles, which are these little gray balls, right? Those are inflating. I'm not positive, I can't remember at the top of my head what the osmotic vacuole inflation mechanism is. I don't know if it's a calcium pump or a sodium pump. It escapes me. But there's some sort of osmotic gradient that causes, and these pumps, ion pumps, just like any vacuoles or any osmotic gradients in cells, right? And that's causing these to inflate. And when they inflate, it's like a balloon animal. That was what I used. Don't screenshot that, please. The balloon animal is what I was using in my last long lecture of how this works, right? So, you know, when they make the wiener dog or whatever, they blow out the balloon and it goes longer like that. It's exactly what's happening here. We're inflating these vacuoles and it's elongating and it's kind of pushing this stretching action that's happening during this growth and development. So along the edge here of the PSM, we have this fibronectin-integrin interaction, which couples the PSM with the notochord. So this is also allowing it to be pulled along, because remember there's this the integrins and the fibronectin integrin those are scaffolding, right? Or glue, kind of like the cadherin type stuff, which pulls the PSM during the notochord elongation. So those three processes, the cell migration, the cell division after migration, and then the vacuole inflation are what drive this notochord elongation. Okay, and so here is just a micrograph of this process. So we have 17 hours post-fertilization. You can see these small vacuoles. At 19 hours, they're bigger. 21 hours, they're even larger. The red arrows, for whatever reason, they are denoting the nuclei. I guess maybe just to differentiate that that's not the additional vacuoles that aren't being inflated. I'm not sure. But those are nuclei, so don't worry so much about them. Worry about the big V's, vacuoles. That was my nickname in college, Big V. No, it wasn't. Okay. So the transverse section through the chunk of the chick embryo on days 2 through 4. So we're starting here at day 2. We have the epidermis. You probably are getting sick of seeing the neural tube and the notochord. We've seen this last few weeks. We have the sclerotome and the somites. So these somites are these little units, like Lego bricks kind of. And at the bottom we have the sclerotome progenitor cells. At day 3, we start to get some differentiation here and some proliferation and migration. The dermatome here is going to be the top, the sclerotome is going to be the bottom. Day 4, we're getting more differentiation. So we see that we have the myotome cells start to, and myoblasts start to differentiate. Myo meaning muscle, right? So we're going to have some muscle. We have the dermatome, which is up on top towards the dermis side. We have the dermamyotome, which is a little bit more lateral to that. So the dermatome is up on top. The dermamyotome is a little to the sides of that. The sclerotomes down at the bottom here, they start to proliferate and kind of migrate out. And then we have, the premaxial myotome in the middle, and then we have the abaxial myotome that's just kind of more towards the side, just kind of anatomy terms for location here. And then we also have this dermamyotome lip, so the folding over occurring there. Day 4. So now we got a bunch of myoblasts again, myo meaning muscle. Central myotome region here. Cells kind of migrating more towards the dermis, through the dermamyotome region. We have this premaxial dermamyotome lip. So this is just that folding region in the abaxial. So premaxial towards the middle, abaxial towards the outside. This is actually a somatic bud now. The lip's been turned or relabeled somatic bud at this point. The sclerotome cells proliferating like crazy down at the bottom here. Dorsal root ganglia. Remember, this is part of our ganglia being neurogenesis or the neural development. This is happening at the same time in between. We've talked about that already, so I'm not going to focus on that, but just kind of giving you a not only location but temporal aspect of how they relate to each other. Okay, so we look at all these wonderful Technicolor, beautiful figures all the time, and so we're going to actually look at a micrograph, kind of see what's going on here. So we have the neural tube and the notochord, right? We have the ectoderm, surface ectoderm, which will be part of the dermis eventually. 5, we have the intermediate mesoderm. 6, we have the dorsal half of the somite, so dermatome and dermamyotome. 7, we have the ventral half, which would be the sclerotome. 8, somitical, so it's kind of the middle at this point of the somite. And that's it. For some reason on my notes I have 9, 10, 11, 12, and 13. I don't know how that happened. Maybe I was hallucinating additional anatomy figures. I don't know. All right, let's go. I told you I'm in all over the place now after having given this lecture just a half hour ago. So, all right. So how does this all work? Gradients. That's always the theme. Like gradients. That's always what causes this stuff, right? It's positional location. So location, location, location. That's what they say in setting up a business, right? That's also for development here. So the destiny of a particular region of the somite depends on the location. So in the case up here, we have the neural tube, and it is expressing NT3 and WNT1. Down at the bottom, it doesn't express those, right? So if you're part of the somite and you experience a lot of NT3 and WNT1, you know to become dermatome or dermamyotome, right? So the red or the intermediate pink, I guess. As you move further towards the ventral side, this concentration gets less and less and less, right? So you don't become dermatome or dermamyotome. Now, this is also something that happens during neural development. There we talked slightly about during neural development is that the notochord expresses sonic hedgehog, and that helps the floor plate development during neural tube development, right? Well, this also diffuses out towards the somites. And so if you experience a lot, if you're part of the somite and experience a lot of sonic hedgehog and noggin, then you know it's time to become sclerotome and not dermatome or dermamyotome, right? The actual action of noggin, so it's not really experiencing noggin. Noggin's actually coming and blocking FGF5 over here, which is from the lateral mesoderm being expressed. So there's also these additional signals. So BMP4 and FGF5 are expressed out here laterally by the mesoderm, the lateral plate mesoderm. So down on this side, noggin's blocking a lot of this, but up at the dorsal side up here, BMP4 and FGF5 are not impeded by noggin, since noggin's not coming from the dorsal side here. So they're able to aid in this development here. And you can see that not everything, like, what I like about this figure is not everything is, we don't know everything about everything yet, right? So you can see here that we have PAX3 and PAX7. The epidermis we know expresses BMP4, which leads to the top of the neural tube development here. But we don't know, there's something missing. We don't know what's causing PAX3 and PAX7. At the point of this lecture, I haven't looked at the literature lately to see if that's ever been uncovered. But we don't know what's causing this expression to change and aid in this process. Okay. So yeah, so that's how we get this differentiation of the sclerotome and the dermamyotome and the dermatome. All right. So I went faster through that because this is kind of where I realized I wasn't recording last time. I'm looking down to make sure I'm still recording, and I am. So let's look at this. So during this developmental stage, we get the sclerotome will start to form vertebrae. And so this is kind of the sequential development of the sclerotome into vertebrae. So remember, the sclerotome is this it's kind of a larger chunk, but is the more ventral chunk as it turns into this triangle or iconic triangle shape of the somite here. You can see that as it progresses, it'll continue to proliferate and build, while like the dermatome kind of stretches out, but the sclerotome really, grows to a point, and then thins itself out. It's kind of like a child, right? So like my daughter, very chunky baby, and now she's starting to thin herself out as she's getting older. So that's what's going on here with the sclerotome, and then it will eventually turn into your ribs, right? So you have your vertebral body right here, your processes of your backbone, your spinous process, and then you have your rib cage, right? Okay, moving on. So in addition, so the segmentation, right? Think if you look at your vertebrae and everything surrounding your neural tube, which is your spinal cord eventually, right? That segmentation happens during the somite stage, but you can see evidence of that now, right? So each vertebrae is like in each spinal segment, right? And then along that we have like myotome tissue that will kind of interface with your nervous system, right? Because they need to be controlled by your nerves, right? Muscles sitting there with no input signal or output signal, or just tissue. They're not doing anything. But you can see that we have our sclerotome, and then we have this these two different segments, so rostral or caudal, so that's just based on the gradients, right? So we have anterior and posterior. But those different segments are preserved and lead to this vertebrae formation. Make sure I'm not missing anything here. So, okay, so one thing I am missing, so the sclerotome is actually, so right here, so we know we have like this differentiated segment. It's actually penetrated by this growing nerve tissue and recombines, right? So you have a caudal segment and a rostral segment, or a posterior and anterior, if you're looking at human anatomy. After it gets divided in two like this, they recombine. So your vertebral segments here, or your vertebrae, are a combination of the top half of one sclerotome and the bottom half of another sclerotome. So, and they indicate that with the blue and the tan, orange, peach, probably peach colored segments there. All right. So let's talk about, okay, so this is kind of going back to the notochord elongation in the vacuole filling. So if you remove the extracellular matrix sheath around the developing notochord, so that surrounding tissue, we mess up the formation of the spinal cord. So it gets kind of like inhibited. So here is this Col8A1A. That stands for collagen 8A1A. This makes it even, doesn't clarify a whole lot. It's a collagen. Anyways, so the collagen is normally expressed throughout the spinal column. And you can see this through this green expression is of this of a collagen. This is 2A1. But a second collagen gene is Col8A1A. So let me take a step back because it's confusing because all these acronyms. So the green here is Col2A1. So it's a different collagen gene. So there's multiple collagen genes, right? So now if you get rid of one of those collagen genes, Col8A1A, you can no longer straighten the spinal cord, and it results in these like fused vertebrae. And so the surrounding tissue the presence of the collagen, and remember, this notochord is tied to the somites through those integrins and things like that. So if you remove some of this that is there during the stretching process, you get malformation of these vertebrae. It just ruins the process. So what happens to the notochord, right? So you don't ever hear someone's like, I've hurt my notochord. Like, when you become an adult, you don't have, it's not called the notochord anymore. So while some of it just disappears, some of it is turned in to nuclei pulposi, Pulposi. I don't think it's labeled here. It is not. Oh, it is on the next slide. Okay. So it's like a gel-like mass in the center of your intervertebrate discs. And it's surrounded by this fibrous tissue called annulus fibrosus, which is a connective tissue that develops from the sclerotome. So the nucleus pulposus in this case is the NP in your discs here. And the AF is the annulus fibrosus, is that fibrous tissue, and then the V is the vertebrae. So you can see this is your vertebrae here, and then in between you have discs. So I was fortunate, sarcasm, to have a slip disc 2 years ago. I remember, so I had a disc, it was one of my C something up here, that was like, it wasn't herniated, but it was like really messed up. And I was teaching, I think I was teaching development at the time, and it was during right before Christmas break, and I told my class, like, because your nerves protrude from there, right? So your nerves come through your near your discs there, and lead to all your peripheral nervous system. We talked about this during neurulation, and I may have told the story already, but we'll say it again. But the nerves go down to your arms or whatever, and so if you have a slip disc, there's pressure put on those nerves, and sometimes it can cause sensation in your arms. And I joked to my class, I was like, I think I'm having the world's longest heart attack, because I had like numbness and like messed up feeling in my arm, which is also a symptom of a heart attack. Ended up being a slip disc, but yes. So within this disc, tangent is what the notochord turns into. All right, I'm gonna try not to be tangential. We're already at 22 minutes. So here's the notochord early. As you can see, we have this notochord sheath and the remnant here in the middle, and then we also start to form these little pulpus in the middle, and so little is known about the nuclei pulposi formation, but it kind of is, appears that mechanical forces squeeze off sections of the notochord. So that's what these red arrows are, that it just kind of gets pinched into these little spots. And it also leads to the loss of proper, so in some of these mutants, it can lead to loss of sonic hedgehog signaling. And if there is this loss of sonic hedgehog signaling, then we get malformed pulposi, but not much is really known. We know what turns into that, but like the kind of the function and formation there's not a lot of knowledge on that at the time of this lecture. All right, sorry, I'm trying to kind of speed through the end here. So tendon formation, so there's also ligaments and tendons that are formed that connect bone and muscle, which is important. And so there is a gene called scleraxis that is expressed in progenitors of tendons, and so you can see that is also arising from the somites here. Just to comment on that. And so this scleraxis is fused to GFP here. So you can see that this is a big component of connecting muscle, or those myotome cells, to the bone cells. So you can see the muscle or the tendons all here, and they connect with those what eventually become muscle, or myotome which eventually become muscle. Um, and so this scleraxis is in the syndotome here. So we have the sclerotome, the dermamyotome, and then in between here we get the formation of what's called the syndotome. The syndotome is the tendon precursors, right? So if you have what's going to become muscles are always connected to bone or other muscles via ligaments or tendons, right? So if we have the development of the bone and we have the development of the muscle, we have to have something that connects them, right? And so that's where we get the syndotome development. It's just a thin layer, and it's just the glue, right? It's just that tissue that connects the muscles to the bone. So plus we have cartilage too that connects bone to bone. We're not going to get too much into this, but there, just to see Sonic Hedgehog again, FGFH again, SOX9. When we get into the last chapter, will probably be sexual differentiation, and we'll talk a bit about SOX9 there. But just know, this is just, I'm not going to get too much into this, but Sonic Hedgehog involved again, of course. All right, and then lastly, so this is the last slide. Like I said, I'm trying to keep it within our normal time frame here. So we have our neural tube developing, the anterior, posterior. We have the vertebral progenitors here, which at the time are, which will become cartilage here in the muscle progenitors. And so you can see the tendon progenitors in between here. Again, tendons important, so they do arise from the somites similarly to the myosin, the muscle precursors and the bone precursors, and then we have our, so you can see kind of they're just positioned in between. It's a preservation of position, right? So they start off in between the muscle, what will become muscle and what will become bone, and then they end up becoming the tendons, which are in between muscle and in between bones. So we have the muscles in between your ribs, and then you have the ribs itself. We have the vertebrae. We'd have the neural tube running up this way, etc. Okay, that is it for this lecture. The last couple slides I kind of flew through. I was debating if I wanted to keep them in or not anyways. Just to make you aware, I'm not really focused on quizzing you hard on that stuff. It's not as important as some of the other stuff. Anyways, thank you for watching. I hope you enjoyed this lecture. I didn't enjoy giving it twice in a row. But thanks for watching, and I will catch you in the next one.